This video is about the microbiota-gut-brain axis as it relates to psychiatric disorders, presented by the modern psychiatrist. The gut is many things, and one of those things is a sensory organ. It has bidirectional communication via nerves, hormones, and inflammatory molecules. By bidirectional communication, I mean there is communication from the gut to the brain, and that's 90% of the communication. Then there's also communication from the brain to the gut. That's 10% of the communication. The enteric nervous system is part of the peripheral nervous system, and it includes a highly innervated neural network dedicated to facilitating central nervous system communication with the gut. The enteric nervous system is the gut's nervous system, also known as the body's second brain. There are 50 to 100 million nerve cells within the enteric nerve system, which is as many as are contained in the spinal cord. There are many types of cells that are found in the gut. One type of cell is immune cells. These immune cells make up the largest component of the body's immune system, and there are more immune cells in the gut wall than in blood and bone marrow combined. There are also endocrine cells in the gut. The total number of endocrine cells in the gut is greater than all other endocrine organs combined. From these endocrine cells, more than 20 different types of hormones are secreted. Serotonin can also be found in the gut. 95% of the body's serotonin is stored in the gut. Within the gut, serotonin coordinates the contractions of the intestines, and within the body, it's also involved in sleep, appetite, pain sensitivity, and mood. The interaction of the gut microbiota and the central nervous system is the microbiota-gut-brain axis. The microbiota is composed of over 100 trillion microbes which live in the gut. There are 1,000 bacterial species and greater than 7 million genes. The composition of the microbiota is determined by several factors. Some of these factors include genes, the maternal microbiota, microbiota of household members, the diet, the brain's activity, and state of mind. The gut microbiota influences many aspects of physiology via neural, hormonal, and immunological pathways. It also contributes to the maintenance of health. The gut microbiota can disrupt homeostatic regulation, and it may influence the etiology and pathophysiology of many diseases, including mental disorders. When the gut microbes fall out of their healthy, stable state, this is called dysbiosis, and it occurs in several gut disorders. Gut symptomatology and mental health are closely linked. Gut symptoms are the most common somatic symptoms associated with depression. Anxiety disorders are the most common psychiatric comorbidity in functional gastrointestinal disorder patients. In the treatment of irritable bowel syndrome, antidepressants and psychotherapy have been found to be effective. Mental disorders are not only central nervous system disorders, but they're disorders with complex systemic interconnections. The gut mirrors our emotions. This has been found to be true in several observational studies. When someone is hostile or aggressive, their intestines contract quicker, versus if someone is feeling hopeless, inadequate, or self-reproachful, their intestines contract slower. The remainder of the video will contain information from this journal article, a systematic review of gut microbiota composition and observational studies of major depressive disorder, bipolar disorder, and schizophrenia. This can be found in the Journal of Molecular Psychiatry from April of 2022. The full reference can be found at the end of the video. This systematic review was comprised of 44 studies. 24 studies involved major depressive disorder, 7 studies involved bipolar disorder, and 15 studies involved schizophrenia. The objective of this systematic review was to compare microbiota composition between cases and controls across multiple mental disorders. Alpha diversity was studied in this systematic review. Alpha diversity is the measure of gut microbiota diversity within a single sample. It describes the gut microbiota based on richness and evenness. Richness is the number of bacterial species present in the gut, and evenness is the distribution. Beta diversity was also studied in this systematic review. Beta diversity is the measure of similarity or dissimilarity in gut microbiota communities between groups. The studies in the systematic review investigated whether people with or without a mental disorder had different gut microbiota compositions. The purpose of the individual studies was to investigate differences in the gut microbiota composition of patients with or without a mental disorder. However, the aim of the review is to determine consistencies in gut microbiota in major depressive disorder, bipolar disorder, and schizophrenia. The majority of the studies reported no difference in alpha diversity between psychiatric cases and the controls. For major depressive disorder and schizophrenia, no difference in alpha diversity was found for the majority of the studies. For bipolar disorder, 15 of 24 studies showed no difference in alpha diversity, but 8 of 24 studies reported lower alpha diversity compared to other disorders. One study reported higher alpha diversity. There were 2 ways that the studies analyzed beta diversity. The first way was statistically, and the second way was by using ordination plots and observing clustering. The review reported results about the bacterial microbiota based on taxonomy. The phylum is the broadest level of classification, all the way down to species, which is the most specific. Here are the bacteria most commonly reported to be different between cases and controls at the phylum classification level. These are the bacteria that differed between cases and controls at the class level. These bacteria differ between cases and controls at the order level. These bacteria differ between cases and controls at the family level. These bacteria differ between cases and controls at the genus level. For patients with major depressive disorder, these bacteria were found in higher or lower abundance compared to the controls. For patients with bipolar disorder, these bacteria were found in higher or lower abundance at the genus level compared to controls. For patients with schizophrenia, these bacteria were found in higher or lower abundance at the genus level compared to controls. In patients with major depressive disorder and in patients with schizophrenia, these bacteria were found in higher abundance or lower abundance at the genus level compared to controls. In patients with bipolar disorder and in patients with schizophrenia, these bacteria were found in higher or lower abundance at the genus level compared to controls. In patients with bipolar disorder and in patients with major depressive disorder, these bacteria were found in higher and lower abundance at the genus level compared to controls. In patients with bipolar disorder, patients with major depressive disorder, and patients with schizophrenia, these bacteria were found in higher and lower abundance at the genus level compared to controls. At the species level, 18 species were found to be consistently different in cases versus controls. Functional potential was the topic of some studies within the systematic review. Functional potential refers to the range of functions that a particular biological system is capable of performing, often used in the context of studying the relationship between an organism's genotype and phenotype. Cell motility and secretion and membrane transport were pathways enriched in controls compared to cases. Major depressive disorder was associated with an enrichment of glycan biosynthesis and metabolism, lipopolysaccharide biosynthesis, and biosynthesis proteins and transport and catabolism pathways. Another study identified 31 pathways associated with bipolar disorder: tetrahydrofolate biosynthesis, pentose phosphate pathway, and ornithine biosynthesis. Pathways associated with controls included peptides and nickel transport systems, branched chain amino acid transport system, and putative sugar transport system. Functional pathways commonly enriched in schizophrenia were alpha-linolenic acid metabolism, ascorbate and aldehyde metabolism, geranyl degradation, nucleotide metabolism, and pertussis pathways. Pathways commonly enriched in controls were phenylpropanoid biosynthesis, RNA transport, and starch and sucrose metabolism. The following 3 studies reported associations between gut microbiota composition and psychotropic treatment. One study collected data at 3 time points across 30 days of escitalopram treatment. They observed changes in bacterial taxa across the 3 time points, suggesting compositional changes associated with treatment. One study reported changes in gut microbiota composition after individualized treatment with escitalopram and found that gut microbiota composition was more similar to controls after treatment. A third study observed changes in gut microbiota composition after 4 weeks and 8 weeks of treatment with 40-oxetine hydrobromide and observed attenuation of the differences in alpha diversity between cases and controls associated with treatment and changes in the relative abundance of specific taxa after treatment. A bipolar disorder study observed differences in the enrichment of specific bacterial taxa associated with 4 weeks of quetiapine treatment. A schizophrenia study observed a reduction in the number of bacteria that discriminated cases from controls after 3 months of antipsychotic treatment. And another schizophrenia study observed an increase in alpha diversity, which is changes in the relative abundance of Lachnoclostridium and Rhomboidsia, and the potential for basal levels of these taxa to predict treatment response after 24 weeks of treatment with risperidone. In the discussion section of the systematic review, it was stated that no strong evidence for a difference in number or distribution of gut bacteria, i.e., alpha diversity, and those with a mental disorder compared to those without one. There were consistent differences in the overall composition of the gut microbiota, which is beta diversity, between cases and controls within each mental disorder category. Previously, higher alpha diversity had been considered a marker of better gut health. The assumption was that greater species number and diversity may increase gut ecosystem resilience and stability due to increased functional redundancy for metabolic functions and a more robust resistance to pathogenic invasion. Growing evidence for gut microbiota research indicates that alpha diversity metrics are of limited utility as a measure of gut health or to discriminate between disease cases and controls. This is further evidenced by another recent systematic literature review and meta-analysis of gut microbiota composition across psychiatric disorders. The authors reported no difference in alpha diversity across mental disorders. Beta diversity analyses consistently suggested that individuals within each mental disorder category harbored a more similar gut microbiota composition to each other than to the clustered compositions observed in healthy controls. The following were limitations of the current review. Geographical distribution of studies shows a clear overrepresentation of studies conducted in China, and different geographic regions are associated with different microbial compositions. This review focused only on bacterial members of the gut microbiome and did not include other microorganisms such as archaea, viruses, bacteriophages, and fungi. The review concluded that major depressive disorder, bipolar disorder, and schizophrenia were not characterized by differences in number or distribution, alpha diversity, of gut bacteria. Each mental disorder appeared to display overall compositional differences compared to controls, which is beta diversity. Identification of lower levels of butyrate-producing bacteria, higher levels of lactic acid-producing bacteria, and higher levels of bacteria associated with glutamate and GABA metabolism was relatively consistent across studies. There is an urgent need for the harmonization of reporting and methodologies in the field of human microbiome research in order to achieve consistency and reproducibility. Thank you for listening. The following are the references for this video.